Mechanisms of Antimicrobial Action, Chapter 10. The learning objectives for this topic are: explain the principle of selective toxicity, list 5 mechanisms by which antimicrobial drugs affect the growth of pathogens, describe the actions and give examples of drugs that affect the cell walls of bacteria and fungi. The topics for this subtopic are inhibition of cell wall synthesis, inhibition of protein synthesis, disruption of cell membranes, inhibition of general metabolic pathways, inhibition of DNA or RNA synthesis, and prevention of virus attachment. Let us begin with inhibition of cell wall synthesis. We have quite a few drugs that inhibit the synthesis of bacterial cell walls. The reason being fairly low toxicity to the patient. We don't have cell walls on our cells. And we especially do not have peptidoglycan in our cells, so we can attack the synthesis of peptidoglycan without causing side effects to the patient. We call this selective toxicity. Let's go over how bacteria synthesize their cell walls. As the bacterial cell grows and basically doubles in size before it gets ready to divide in two, binary fission, subunits of NAG and NAM are added, and they're cross-linked with tetrapeptides. All of this is going on while the cell is growing. Makes sense. As you're enlarging the cell, you've got to add more to the cell wall. Beta-lactams inhibit the formation of these cross-links. That includes penicillin G, which is the natural penicillin, and then the semi-synthetics that we have at our disposal. So what happens is, since the penicillin goes in and inhibits these cross-links, as the cell grows and tries to add more NAG and NAM units, it starts to get holes in the wall, and various assorted ions and sugars can leak out of the cell and the cell dies. That's the main function of your beta-lactams, like penicillin. Now, there are other cell wall synthesis inhibiting antibiotics that function in a little different way. Vancomycin interferes with allene-allene bridges, and those are some of the amino acids that are involved in this tetrapeptide. So they're a little more selective in interfering with the cross-linking than penicillin is. And because vancomycin isn't a beta-lactam, it is immune to beta-lactamases. So often we'll put patients on vancomycin if they have a microbe that is resistant to the beta-lactams. Methicillin-resistant Staphylococcus aureus, or MRSA, is a candidate for vancomycin. Bactericin, this shows up in antibiotic creams all the time, and this blocks the transport of subunits. So as the cell is growing, it gets fewer and fewer bricks, and that creates the holes. Now for our last one under the topic of inhibition of cell wall synthesis, one that is specific for acid-fast bacteria. Acid-fast bacteria grow very slowly, and they're very hard to treat with, say, penicillin, vancomycin, some of these other things, because they're growing so slowly. The holes form slowly and the cell can find ways around it. So we have a tendency to use isoniazid, which disrupts the formation of the acid-fast layer, disrupts formation of mycolic acid, of which mycobacterium are named. And this has a tendency to work better because, one, it's not only forming holes in the cell wall, but also that acid-fast layer makes the cells difficult for white blood cells to phagocytize. So if you disrupt the acid-fast layer, the cells are more easily phagocytized by your immune system. Unfortunately, because acid-fast bacteria grow so slowly, you have to be on isoniazid for between 6 months to a year, and that becomes a problem for a lot of patients. Next up, inhibition of protein synthesis. We have several antimicrobials that target 70S ribosomes. And because we're targeting 70S ribosomes, a lot of times they leave 80S ribosomes alone, and you have selective toxicity again. So let's talk about some of the groups and how they inhibit the 70S ribosomes. Aminoglycosides cause a change in the shape of the 30S subunit, and that causes the mRNA to be misread. So you get incorrectly formed proteins, which cause the cell to die. Tetracycline, which you may have heard of, and some aminoglycosides block the docking site of tRNA. So you can't bring in more tRNAs, you can't lengthen your developing protein. Chloramphenicol blocks the active site where the two amino acids are joined together. So once again, you block protein synthesis. Lincosamides and macrolides bind to the 50S subunit so that it can't move along the mRNA, once again inhibiting protein synthesis. Now, antisense nucleic acids bind to the mRNA, so it's a two-stranded mRNA, and the two subunits can't clamp over the top of it. It's too big of a mouthful to get the mouth over, so to speak. And then our last one, this blocks where the 50S subunit would clamp over the top of the 30S, once again inhibiting protein synthesis. On to disruption of cell membranes. Now, we don't have very many antimicrobials that disrupt cell membranes because in all cells, well, most prokaryotic cells, archaea, have a different cell membrane. But for most of us, we have this phospholipid bilayer, and we have pretty much the same stuff in it. So if we're going to selectively attack the cell membrane, we've got to look for molecules that are unique to the pathogen we're trying to kill and are not found in human cells. So one of these is ergostol, and it is in the same class with cholesterol. It's a little bit different. So we have a drug, amphotericin B, that binds to ergostol, causes them to line up, and this causes a pore to form in the cell membrane, and we can have leakage out of the cell, and it causes the cell to die. Now, amphotericin B preferentially binds to ergostol, but it will bind a little bit to cholesterol in human cell membranes, so you have to watch amphotericin very carefully. Now, we've got other drugs that block the synthesis of ergostol, and they have fewer side effects. Next up, inhibition of general metabolic pathways. Now, in inhibiting general metabolic pathways, you have to pick pathways that are unique to your pathogen. You don't want to attack glycolysis because that'll attack the cells of your patients. You don't want to attack the Krebs cycle, you don't want to attack electron transport chains, that kind of thing. So when we're attacking metabolic pathways, we got to look for stuff that's unique. The very first antimicrobials developed were arsenic, mercury, and antimony containing antimicrobials, and we don't use them today unless we've got absolutely nothing else to treat the pathogen, because they do inhibit enzymes, but they also inhibit your enzymes. So the plan is with these is that we're going to inhibit and kill microbes that are growing faster than your cells. It's kind of the same concept of chemotherapy. So let's talk about the first widely used antimicrobial, the sulfa drugs, and they attack this metabolic pathway. Basically, we take a precursor molecule, PABA, and it goes into this enzyme, combines with two other substrates, and we get dihydrofolic acid. Then we turn that into tetrahydrofolic acid. From there we can make purine and pyrimidine nucleotides, and from there we can make DNA and RNA. Now, we humans do not have this enzyme. We cannot make dihydrofolic acid. We have to consume folic acid in our diets so that we can go from here to here to here, so to speak. So this is a good pathway to attack. So here is what PABA looks like, and you can see how the sulfa drugs have this same benzene ring with an amine group, but we've got different things over on this end other than this acid group. But they fit into the active site of the enzyme just fine, blocks the entrance of PABA, and we don't get hydrofolic acid produced. Leaves our cells alone because we're importing folic acid. But this shuts down indirectly nucleic acid synthesis. Now, what kind of inhibition would this be? That's right, competitive inhibition. There are some antimicrobials that inhibit DNA and RNA synthesis directly at DNA and RNA polymerization. Here's one of them, acyclovir, that you may have heard of. We give this to patients who are HIV positive because as their immune system drops, herpes viruses will come out of latency and start causing them problems. We also use this for patients that have severe shingles, which is chickenpox virus coming out of latency, out of your nerves, and they can cause severe problems. So it is an analog, it is a nucleotide analog of guanosine. Acyclovir has the same base as guanosine, but it doesn't have the full sugar. But DNA polymerase and RNA polymerase see this, put it into the line, but they can't hook it to the next sugar because there's no sugar there. So it inhibits DNA and RNA synthesis. Here's another one, AZT. This is also often given to patients with chronic viral infections. This one is an analog of thymidine, but you'll notice instead of an OH, it has 3 nitrogens here. So this one's even better than acyclovir. The DNA or RNA polymerase sees the base, puts it into the line, but because there's no OH there, can't hook it together with the next phosphate to form the sugar phosphate backbone. Now, those are 5 ways of inhibiting microbial growth. We could stop there. That fits the learning objectives for this topic. But we want to talk about preventing virus attachment. These are some relatively recent antimicrobials that work on basically the same principle as a vaccination. Vaccination causes the body to produce antibodies. Antibodies cover the receptors of the viruses so that they can't attach to the host cell. But if you haven't had a vaccination, it's nice to have something that we can give the patient that will prevent that. So oftentimes these drugs are peptide or sugar analogs of the attachment or receptor proteins. So they either are analogs for the receptors on the virus or analogs for the receptors on the host cell. And so these, we've developed them for the picornaviruses, which are polio, which we're currently having an outbreak of polio in Syria, which is really very sad because we just about have polio under control. Also in this family is hepatitis A. So if you're suffering a severe Hep A infection, we can administer some of these attachment antagonists and help you recover from the disease faster, which is the whole point of antimicrobials. Well, that's it for this topic. So here are reminders of what you needed to learn from this topic.